Hi, I am Alyssa Enny, and this is our podcast called Chasing Our Origins. If you have ever been to a museum or have seen one of the many cheesy movies set in some sort of prehistoric time, chances are that you've been exposed to a reconstruction of a past environment. We are often interested in reconstructing past environments as a means to better understand what the world may have looked like many, many years ago. Unfortunately, we have not yet figured out a way to travel back in time to observe these past environments firsthand, but researchers interested in a discipline known as paleoecology have discovered ways to combat this limitation. And no, we're still not talking about a real time machine. We have a researcher here today who uses paleoecology to focus in on some of the most exciting times during human evolution. Her name is Maryse Biernat, and she is a fourth year Anthropology PhD student at Arizona State University. She is affiliated with their Institute of Human Origins and is studying under Dr. Kay Reed. She uses paleoecology to help us gain a better understanding of the environment during critical periods of human evolution, and also how humans may have navigated and exploited the resources on the landscape they lived in. Here is Maryse to talk more about her research in paleoecology. Welcome, Maryse, to our podcast here. So, you had previously performed research that focuses on paleoecology, specifically during time periods of the evolution of our human ancestors. Can you tell us a little bit more about what paleoecology is?
So paleoecology is the study of ecology, but in the past, hence the paleo prefix. Essentially, what I'm interested in is reconstructing the ecology of a certain period of time. This is done using biotic factors, so animals or organisms on the landscape and how they interact with one another, and then also even some abiotic factors such as nutrients in the soil or climate, temperature, things of that nature.
Okay. And so, how do you do this exactly? I know I've heard the term proxy used before. So, what are proxies and how can they be useful in paleoecology?
Yeah, so as you mentioned in your introduction, we can't go back and observe the past, right? So in modern ecology studies, researchers go out and they observe what's happening in real time. We don't have that luxury. So a proxy is a signal that we can see in the past that helps us reconstruct and figure out what was going on. In modern studies, we can go out and look, see this antelope is eating grass. So essentially, what I do and other paleoecologists do is they'll use a proxy to determine diet. So one of those proxies for diet would be looking at enamel isotopes. So the way that plants have a biological signature that gets incorporated into the enamel of the organism that eats it. And by analyzing that signal, we can figure out what types of plant matter are being ingested. We're also left with a very fragmentary fossil record. I think that's really important for understanding why we need proxies, in addition to not being able to observe in real time. So, taphonomy is the study of the transition from the biosphere, the living world, to the lithosphere, which is like the geological world. So, essentially, you have organisms on a landscape, they die, and at each step between death and recovery by a researcher and study of the elements of a fossil bone, there's basically a filtering effect that happens at each point. And so more and more information is lost. Let's say an animal dies of natural causes on a landscape. Scavengers will come in and eat the carcass, which it can be destructive. Some of your smaller scavengers will actually take bits and pieces away. So you'll have hyenas dragging like a leg away. And so the skeleton can be scattered across and not just be left in one place. And then once these bones have been scattered, it then needs to be in a situation where it's buried quickly, because if it's left on the landscape, UV radiation from the sun and just weathering in general can help to degrade these bones. So it has to be in the right situation to be buried, and it also needs to be buried like fairly quickly to mitigate these effects of weathering. And then only once it's been buried can it actually start the process of fossilization where, the bones become mineralized. And that takes a very, very long time. Even after they've been fossilized and they're in the ground, they then need to be recovered. And so, it's all great if there's fossilized bone under us, but if it's 1,000 meters under the surface, it's very unlikely that we're going to find these to be able to even study them. So areas that have high erosion, like the East African Rift Valley, there's a reason why these areas are very productive for fossils, because you have erosion happening and these fossils are coming to the surface and and being exposed, and then we can actually pick them up. And so, you can think of a modern community having all these different individuals of different species running around, but in the fossil record that we study later, it's just little bits and pieces of individuals that are left.
And what kind of time periods do you focus on, and why do you focus on those specific time periods?
So I'm interested in the Pleistocene, mostly because that is where a lot of the action for human evolution is happening. So my previous research has focused between 2 to 1.58 million years ago, so that's solidly in the Pleistocene. But more recently, I'm moving into the Pliocene because that's where we have the appearance of the genus Homo, and I'm interested to see how when Homo comes onto the landscape, how does that change the mammalian communities at the time?
Where do you usually conduct your field research, and why do you focus on those specific locations?
I've primarily done field work in northern Kenya, so at the Kubi-4 Formation, which is to the east of Lake Turkana. But more recently, I've started doing field work up in Ethiopia in the Afar region. And so that's where I started getting my ideas for my dissertation looking into food webs, because that is where we have the first appearance of the genus Homo.
What kind of methods do you use in your research?
So I guess I should just start at the beginning of how we collect paleoecological data to begin with. So a lot of it happens through field-based surveys. So essentially we go out to a locality where we know there are fossils and we know how old they are, and we just walk across the landscape picking up bits of fossilized bones that we find. And so we have a protocol of what we pick up and what we leave behind. What we do pick up is a lot of teeth, horn cores, and then particular parts of postcrania. So with teeth, we can get high confidence to either the tribal or genus level for an identification. Anything lower than that, like if you want a species identification, that requires a specialist to look at teeth, but they are really informative about what exact organism we're looking at. And I'm going to be talking specifically about mammals. Horn cores are found on only horn species, so bovids, which are your antelope. And so the morphology, the way that the horns come out of the skull and the way that they're formed, are able to identify those individuals to species. Any dense bones, which is why teeth and the horn cores are preserved really well, they survive the fossilization process a lot better. And so we pick all these elements up and then we try to identify them down to at least the tribe, if not the genus and species. And so from there, just knowing exactly what organisms, what mammals are on the landscape, we can reconstruct what the community looked like. And so we can also look at the specific elements that we picked up. So for teeth, those are really great for diet. When you eat plants, you incorporate a chemical signature that gets placed in the teeth. And so when you analyze that using mass spectrometry, you can say how many of different types of plants. So generally, especially in the East African landscape, we have C3 plants, which is your shrubs and trees, and C4 plants, which are your grasses. The proportion of C3 versus C4 signal that you read on a tooth will tell you how much that organism ate during its lifetime. So essentially to do that, you drill some of the enamel powder using a dental drill that you would find in every dentist's office. So for bulk sampling, you just kind of go at whatever crack is any defect in the tooth that's occurred during fossilization. That's where you want to drill because you don't want to cause any more damage to the tooth than has already been caused. The bulk sample just tells you what the individual overall throughout its life has eaten. So when you collect the powder, it gets treated before going into the mass spectrometer. And then once it's in the mass spec, then it'll give you a signal of C3 versus C4 vegetation.
So, what kind of limitations do you have using these different proxies in your research? And is there any way that we can try to overcome them?
Yeah, so each proxy comes with its own set of problems and negative biases. So isotopes, carbon is really great because it gives you what the vegetation that organism is eating, but there's a lot of talk about do the proportions of C3 versus C4 that are being consumed by the mammals, is that actually reflecting what was available on the landscape. That's something that, researchers are grappling with. So that's a limitation of that. And, so you can go on and on with all of these negative implications from all of these different proxies. But I think one of the best ways to combat this is by taking a multi-proxy approach. So by combining all of these proxies, you're able to see what is actually going on the landscape during the time period that you're interested in. You're able to, there's some overlap, one proxy might miss out on one thing, but another proxy might reconstruct that in a better way. By putting them all together, you get more pieces of the puzzle, and so you can get a more complete picture because, like I've mentioned, we can't just, walk outside and just see what these organisms were doing.
So, just zooming out for a second here, I want to talk about the broader implications of paleoecological research. What would you say that the goal is of the research that you do?
I'm generally just interested in, what do these communities look like? So I'm interested in all the species of mammals that were present at any given time during the time frames that I'm interested in. Yes, there is a focus, and what really ties it all together is looking at human evolution, but I view these early hominins as just part of the community. And so generally, I'm just really interested in how they all coexisted on the landscape, how they would have interacted with one another, how they would have, been modifying their environments and adapting to their environments through time. And as we have hominins evolving through time, how do they possibly disrupt the communities? There's always been a really big focus on meat eating for hominins. When did it start? Why did it start? How did it start? Were they hunting versus just scavenging? Were we always carnivorous meat eaters? Or did it only start more recently? And so by looking at the community structures, we're able to get at some of those questions that are more human focused around meat eating.
So, where would you like to take this research in the future?
So, as I mentioned, I think that multi proxy research is a really good way to get a better picture of what was happening in these past environments. So, one of the ways that I would like to do that is by studying food webs in the fossil record, specifically around the emergence of our genus Homo on the landscape. And so I would like to reconstruct food webs, and this would be done using multi-proxies. So I would, use these functional traits of, how they diet, what they were eating, and locomotion, how they were moving around. I would take into body size so we could understand who was eating who. That would all go into a food web, and basically I would like to reconstruct the food web before Homo appears on the landscape, when it appears on the landscape, and then after, and just analyze how the food web may have changed through time. Do certain species start to decline, or do they change in any way? And then, if so, could it be possible that they are changing because Homo has now appeared and is a new component into the food web that might be disrupting what was seen before?
So, if you had to give one takeaway message from all the research that you've done, what do you think that that would be?
So my takeaway is that multi-proxy is the way to go, and not just multi-proxy, but interdisciplinary. And so just, keeping current with what other researchers are doing, bringing them into projects so that the multiple perspectives and multiple backgrounds and multiple expertise to be able to get at what is happening in these past environments. It does create a messier picture when you bring in all these different proxies, but at the same time, we're getting closer and closer to the truth. And even though it will be really nice to, paint human evolution with, one broad stroke of what's happening, I personally think that it's more important to get these little nitty gritty details and realize that each place was probably experiencing had different ecologies that would impact human evolution in different ways.
That seems to be all the time that we have left today for our episode. Thank you, Maryse, for joining us. Again, Maryse is a PhD student at Arizona State University. Please join us again next week for another thrilling episode of Chasing Our Origins. Have a great week, everybody.